A paper was published in 2018 in the journal eLife about the mortality rate in populations of captive naked mole rats, a nice little rodent that shows no obvious sign of aging, even when it reaches the canonical age of 30 years. A considerable lifespan if you compare it to its close cousin, the guinea pig, whose maximum lifespan is about 12 years. The paper was published by 3 researchers employed by Calico Life Sciences, the very secretive lab whose goal is to tackle aging and which was created and is funded by Alphabet, the company behind Google. The abstract of the paper reads, "We compiled and analyzed a large compendium of historical naked mole rat lifespan data with more than 3,000 data points. A substantial portion of the population survived at 30 years of age. Moreover, unlike all other mammals studied to date, the age-specific hazard of mortality did not increase with age, even at ages 25-fold past their time to reproductive maturity. This absence of hazard increase with age, in defiance of Gompertz law, uniquely identifies the naked mole rat as a non-aging mammal. In other terms, the fact that individuals composing a population are aging causes the number of deaths to increase exponentially with age. This is called Gompertz law. If, on the contrary, the number of deaths does not increase with age, then this is a serious sign that the individuals composing the population do not age. Some species, called non-senescent, do not age. Could it be the case of a mammal? The consequences would be astounding. A large share of the biology of mammals is the same across any 2 species. The naked mole rat is just a few genes away from the guinea pig or even the mouse. The conclusion would be that some of these genes can make such a dramatic difference that a mammal does not age anymore. In the end, all this sounds like it is not that hard to tackle aging after all. Let's look into the study itself. If an observer started in 2020 with a population of 3,000 newborn naked mole rats, in 2050 we would reach conclusions about what happens to them when they reach 30. The observations could be summarized in what is called a KaplanMajor curve. KaplanMajor curves look like that. This one represents survival rates with time for 2 populations, one who got a flu vaccine and the other one who did not. It shows that for the population of this study, the flu vaccine is associated with improved survival. In our present case, we could draw a KaplanMajor curve by following the whole population of our colony and make the curve go down 1 step each time an individual dies, whatever the cause. So suppose that no individual dies. The curve would be a straight line, like that. Now, this is how the curve would look like if an exceptional storm drowned the colony at some point in time, or if a deadly infectious disease spread in the colony, with some survivors. In such a colony, like in a population in general, this is generally not how things happen. Rather, a series of minor events strikes individuals of the colony here and there from time to time, like, say, a fight between 2 males. The curve may then look like that. This curve represents a population that is not senescent. You cannot see any pattern associated with time, whatever the duration of the experiment. Of course, individuals still die, but individuals die at the end of the experiment in the same proportions than they die at the beginning of the experiment. There is no additional effect of intrinsic causes of death, that is, aging. See the video on the possibility of dying of old age. On the contrary, when a population is senescent, age is associated with the number or impact of such random events. The curve can, for instance, look like that. On this curve, there is a mean tendency of the number of deaths to increase in the same segment of time, say 1 year, even if individual events appear to still occur randomly. Now, the real study on naked mole rats was led on a colony created in the US in the 1980s, that is, around 30 years ago. The KaplanMajor curve actually looks like that, roughly. During the 15 first years, the number of deaths per unit of time seems constant. Then, approximately where the dashed line is, it suddenly stops. Then, around the 80 years milestone, there is a sudden drop. Typically, some event has occurred. Then there is no observed death in the remaining 60% of the colony until it reaches 30 years. This fact can be represented in the form of the evolution of mortality rate with time, and thus compared to a typical Gompertzian curve of mortality rate with time. This is striking on this figure, taken from the study. In short, the curve of mortality hazard through time is not Gompertzian in the case of the naked mole rat. Should we conclude that naked mole rats don't age, where actually there are two caveats, a minor one and a major one. The minor caveat comes from what is called censorship. Indeed, in such a follow-up study, there are dropouts. Some individual naked mole rats are sold or given, others are sacrificed for scientific observations. They do not have any impact on the curve, because it represents the evolution of a proportion. Let's get back to the curve. In fact, the dashed line in the middle of the diagram represents 4 censorship events, leaving 48 survivors. What this means is that the number of individuals in the beginning of the study was 3,000. Then, at day 5,305, there is still 65% of them, that is 1,950. If all individuals were then sold the next day, but 48, among which none died, the curve would still indicate 65% of survivors. On a KaplanMajor curve, a censorship event is represented by a small vertical line. On this figure, it is purple. If a dishonest scientist wanted to manipulate a KaplanMajor curve, they would just exclude individuals they think are about to die. This would artificially improve the curve. Some pharmaceutical companies have been condemned for such practices. There is no reason to think that this is the case here. However, this reduces the combined impact of the two claims that a colony of 3,000 individuals was observed, and that at the end of the experiment, after 30 years, 60% were still alive. Actually, the observation is not on 3,000 individuals for the whole duration of the experiment. The second, more concerning caveat deals with the actual structure of the experiment. It was emphasized by a commentary article by Daman and colleagues, published the same year in eLife. Actually, the follow-up started in the 1980s, but not 3,000 individuals were born then. Most are actually much younger. In other terms, the experiment is not done on 3,000 individuals born 30 years ago, but on 3,000 individuals born in the last 30 years. Then, by a simple translation, their lifespan is represented as if they were all born the same day. This figure from Daman's paper shows the number of animals per birth year. Well, the vast majority of them were born only in the last 8 years before the end of the study. This means that if we combine all data in a KaplanMajor curve, the first 10 years of the 30-year curve represent a high number of individuals. But the last 10 years represent a small number of observed animals. In the actual study, only 23 animals were observed for 18 years or more. This, of course, seriously limits the interest of the study. Imagine one takes a human population and conclude from a small subset of supercentenarians that humans actually do not age demographically because they show no increase of mortality with time. Well, this is not exactly what happened here. But it may have happened for some reason, given that these 23 individuals represents actually less than 1% of the total data points in the study. Daman's conclusion is cautious. This study has actually not really shown that naked mole rats are a non-senescent species. It is just very premature to say so. However, it is true that the survival rate of naked mole rats at age 18 is already remarkable, given that they are sexually mature at the age of 6 months. This is very atypical of mammals. Besides, the fertility rate of breeders does not decline in this interval of time, nor does it decline for the few longer-lived individuals observed. Importantly, most major causes of death, frequent in all mammals, such as cancer, are very rare in naked mole rats. There is definitely something special with these mammals. Yet they are not the astounding exceptions to the law of aging some claim they are. A study in 2011 confirms that they have an exceptional physiology, but also shows that they lose muscular mass at an advanced age and show sign of what could be interpreted as osteoarthritis. In other terms, the picture is more consistent with the hypothesis that naked mole rats are exceptionally slow agers when compared to their close cousins, than with the hypothesis that they are like mammalian hydras or sponges, two examples of non-senescent species.